So we are going to try and do a couple of things. These couple of things are going to include a little bit of R, just a tiny little bit to get those of you that don't know R started. And for those of you that already know R, this is going to be a little bit basic at some point, but I hope that the approach is going to make sense. Okay, let me just quote this real quick. All right, so did anyone have a chance to do what we discussed last time? Okay. So Prerna, do you want to tell us what you did? Okay, so last time we talked about doing a few things like finding a data set and kind of preparing a few slides about it. So I just wanted to see how many of you actually did that, so I have a sense of how many of you are going to be able to say something about a data set that is interesting to you. Did anyone else except for Prerna? Okay, Michael did it. All right, great. So a few of you did. Excellent. So just making sure before we run into things. Okay, so welcome back everyone to this session. We're going to record this session and later on share with everyone. Today we are making progress with our plan. We already talked about next generation sequencing and what the microbiome is. We also talked about how to do some basic mapping on databases. And today we'll talk a little bit more about how to visualize that data and what's in our data. And I wanted to make it more relevant to each one of you so that we can actually start looking at projects that you plan to be working on, that you are interested in, that you would like to understand from both a biological and a bioinformatics perspective. So that's the reason why last time I asked everyone to start thinking about some of the data sets and exploring them similar to what we did last time on NCBI. Now, we want to get to that right away, but let's just briefly review some of what we did last time and answer some of the questions that some people had about mapping of reads and things like that. So last time we talked about metagenomic sequencing, how we can run a sequencer to understand what do we have in our collected sample. And there's a few steps that you kind of go through before that data is prepared. And that method of preparation is going to define how you analyze. So some things are just not going to work. Some questions are not possible to answer with specific data sets. But there are some ways around it that we can also discuss today in the context of today's session. So we already know where to go and search through our activity. I'm not going to go through that again, but we're going to just briefly see how the different elements come together. So we already looked at the courses. What we are doing in the practice sessions is we're using an example from Metagenomics 1. That's a course. So if you had a chance to practice what we did last time, you would find all the relevant information in Metagenomics 1. What we did last time was also a demo pipeline. That demo pipeline quickly produces outputs because it's already run, but it's also kind of demo data. So to really understand how to run those data sets, I hope that you had a chance to go through the tutorial in Metagenomics 1, because if you want to analyze data sets that you have found on NCBI, today I will show you how to actually download the data from NCBI and prepare that pipeline. You have to understand how all the settings come together, and so it's useful to do that tutorial in Metagenomics 1. There are also scripting challenges or some things that are just easier to do using scripting. So we're going to briefly look at that today and hopefully just do a little bit of an introductory kind of a lesson that all of you, I hope, can complete, understanding how this fits in to the overall program. Now, we started talking about this human microbiome project. We reviewed it already, so I'm not going to go through that again. And we all understand that the idea of this relationship that we are looking inside the microbiome has to do with the relationship between genomic sequences of a particular gene. So which gene, let me briefly ask you, which gene helps us organize our present day species, or the species that we observe in a sample, by taxonomy? Which gene are we using? All right, excellent. Yeah, 16S ribosomal RNA. Now, is this the large subunit or the small subunit of the ribosome? All right, great. Now, does anyone remember which database we are using to find those relationships, the taxonomy, to annotate the taxonomy? Okay, we can use NCBI or Silva. Excellent. Okay. So as you remember, the way to prepare this is to take a sample, and once we have the sample, the DNA or RNA is extracted. Then after it's extracted, it's shattered. This could be a mechanical kind of physical shattering, or it could be using some enzymes. And then once the sequencing happens, it depends on what kind of sequencing is actually done, but those are short reads, and those short reads have to be mapped onto some reference. And so that's where these databases come very handy. And in those databases, we typically use a different set of references. So we can use, for example, on NCBI, we have the full genomes, all bacterial genomes. That's a lot of genomes. Or we can use just the 16S. Now, the 16S is useful because different levels of taxonomy could be assigned based on the different hypervariable regions. There are 9 hypervariable regions, and we know which sample or which kind of organism within the sample belongs to which group by the different levels of taxonomy, by mapping and counting how many reads actually align to a particular place. So we looked at an example of alignment, and there was a question. The question was, how do we know how reads align to the reference genome? If I'm correct, that's how I understood the question. I don't exactly remember what the actual question was, but I believe it was to just reiterate and explain again how the reads fall onto the reference genome. So each one of these lines right here represents a read. And the set of reads that we originally have, this is shotgun sequencing or amplicon sequencing, it doesn't matter. We only have a small portion of reads, or actually a large portion of reads. So we have a lot of short sequences. And the goal is to understand, where do they map? So if you imagine, in this case, we used one single genome as a reference. We can, in the same way, organize all of the 16S genes one after the other and just use it as a reference genome and look, where do our reads fall? Where do they align well? So here you can see that by matching the sequence, so the T to T, A to A, G to G, C to C, etc., and allowing for some flexibility. For example, A to C doesn't match, but because so many of the other ones do match, we say that this aligns to this portion of the genome, and we can accurately estimate the position, the beginning and the end of either the mapped portion or the whole read. So based on these positions of beginning and end, we prepare a new file. That new file contains all of the reads organized by the beginning and the end of those positions, including some of the variation that we see in the mapping quality. So this mapping quality, if we have a full pipeline that we designed, we actually can play around with the quality, because it's obvious that some sequencing errors will be there. Also, there will be some other issues that we will discuss in our next lecture, where we will look at you know, how we can overcome this challenge with with mapping quality that essentially is not ideal. Okay, so here, does anyone have any questions about this process? Because unfortunately, I don't remember who it was that had the question, but just making sure that I'm addressing that issue that we had. Okay, all right. So again, if someone had a particular question about the mapping of reads, I'm happy to answer that. I just do not remember exactly who it was. So let me see really quickly. Okay. All right. So another thing that we looked at are the different databases. So let's, okay, J-Deb. So J-Deb, are you here today? Okay, J-Deb, I see that you're here. If that wasn't clear, please let me know. So let's take a look at this database, Silva, together. To get to Silva, we have to go to Silva. So this is the link. Okay, so please navigate to the Silva database. And let me know once you're there. Okay, so Silva actually has a number of different things that you should probably know about. One of them that we are using is the actual database. So this database, what's good about it is that it is quality checked and aligned ribosomal RNA sequence data, right? So there's actually people checking this and updating this database all the time. Now, there are some things that you can also look here. They have some web applications, and they also have an actual software package. And the software package allows you to do all kinds of analysis, which is good. Now here, let's go to search. Well, actually, sorry, let's go to browser. Okay, so if you're here, you can see by the different levels of taxonomy, you can see, for example, in Silva, bacteria is the kingdom, right? So if you go to bacteria, now you have different levels of taxonomy, okay? So here, once you do in the quick, in the levels of taxonomy, you will see that we can select one of them, for example, actinobacteria. And here we can select, again, actinobacteria. And for example, let's select, I don't know, actinomycetales, okay, and this one. And so here you can see that if you were to download, let's say you want to do this on your computer, right? So if you wanted to download this actinomycetales and you want to add them to the cart, you can see that it contains 523,912 sequence entries. So if you add them, okay, just adding these sequences to your cart right here, it will take some time, but I just want to show you what this would look like. Okay, anyways, it will take a little bit of time because this is over 500,000 sequences. What is actually inside this data set? So inside this data set, you will find a FASTA file. Let me quickly, okay, there you go. So you can see that 523,000, obviously this is a lot of data, so we're just going to clear that. I just want to do this a little bit quicker. Let me see. All right, let's clear it. So let's go to this one, and I'm just going to select the Actinobacterium. Okay, and here there's 57. So I'm going to add these 57, and I can click on download so that we can see what's inside. Now, if you want to download this, it can either come with gaps or without gaps. And so let's do without gaps for now. And click on Start Export. Okay, let's see. Okay, so I think I just did this previously just to make sure that everything works. And let's see what's inside. Okay, so here is my download. Okay, so what's inside is actually a single FASTA file, right? So you can see that this is .fasta. And here it says for each one of our entries, so there's 57 of them. If you do search for this symbol, you will see, okay, there's actually 61 of them here for some reason. I don't know why, because it was supposed to be 57. But you can see here that each one contains a sequence, and this sequence represents the 16S gene from this particular organism or species, depending on different levels of taxonomy that are known for this particular 16S. Now, when we are doing our mapping, as you saw, it doesn't have to be perfect. And the reason for that is because we have a whole variety of unknown organisms that we just don't have any annotation for them. So what we can do is we can use this flexible mapping so that we allow for some mistakes. Let's see, so this is, imagine that this is the sequence, right? And you have a number of reads. So what you want to search for when you are exploring the way the data set is prepared are a number of things related to the mapping. For example, you want to know the average read length. You want to understand how the data was prepared. Was it paired end or single end? And you typically want to also know which instrument was used to prepare the sequencing. That allows you to understand the level of error rates that you are expecting in your reads. Okay, now we do have some other databases as well. You can, in the same way, explore some of these other databases. So you can see GreenGenes. On NCBI, you will have the same thing, but right now it should be fairly clear that you have FASTA files as your references. Inside those FASTA files, you already have some annotation about those FASTA files, and you have the actual sequence that you map the reads to. So if anyone has any questions about that, please do let me know. Now, last time we looked at this simple pipeline, all it does is it takes those reads, maps them onto the reference database, counts how many do I have for each one of my organisms that were found, and then replaces those names. And as a result, we got this kind of a table. This table has the entries from the database, and here it has the annotation, and here's the count. Okay, so let's briefly take a look at that table together. Okay. So does everyone still have this table from last time? Please let me know how many of you have this table. Should we repeat how to get this table? Okay, well, I see a lot of you are saying no, so let's just make sure that everybody does have this table, because this is what we want to work with today. So if you go to server.t-bio.info, please let me know once you are here. And when you're on this page, just open up this drop down and it will say Demo Metagenomics Regular. So please let me know once you're here. And for those of you that already have this table, I'm sorry. So did anyone get here to this page? Okay. All right, great. So it's okay. We'll just do this together again. Just want to, okay, don't have the account. So Beepsa, if you could please take a look. Kitty does not have account. We should definitely do that. In fact, I can do that right now. Okay, anyone else? Okay, Kitty, if you could please just put it in the chat box here, the email address that you use. Okay, great. Okay, so what do we do here? We go to Demo Metagenomics Regular. So if you can just click inside here, let's just rebuild this pipeline together. We click on Start. As you can see, we are taking this particular sample, ERR1250035. It is a sample from the American Gut Microbiome Project. We align it to the Silva database. We just looked at that Silva database. You know what's inside. Here you have counting how many reads fall onto each one of our elements. And we replace the name, so we add the annotation from the FASTA file inside our table. Click on End, and download pipeline output files. Okay, so this is the one that we want to get is Metagenomics 1 test table after annotation. All right, so how many of you have the table? Okay, please, just so that we have at least a few people that already have the table. I know that Mikhail has the table. Who else has this table downloaded? Okay, Cam, Prerna, all right, 4, one more. All right, excellent. So let's just open this table in Excel. Okay, so just open it in Excel right now. We want to quickly take a look at what's in this table and try to organize this information in some useful way. So please open it in Excel or Numbers or whatever table manipulation software you have in R. Just open this table. All right, and when you have it, please let me know. Did anyone create this, open it up in any? All right. Anyone else? So I see 2 people. Did anyone else? Okay. All right, so what can we do with this table? First of all, you can see right here that we have K, P, C, O, F, etc. So what does this stand for actually? So if you remember taxonomy, you have the species, genus, family, order, class, etc. Right? So this is the taxonomy. So we can now organize our individual kind of organisms based on this taxonomy. So how can we do that? Well, what you can do is you can take all of this, just let's quickly do this in Excel, just so that we have later on, we'll also refer to this in R. But right now, if we just take this table, right, it's this column, sorry, and you can go to data and you actually text to columns, and here you can do separation by underscore, okay, and you can also treat consecutive delimiters as one. Okay, so here what we want to do is we want to just separate. This is the title and this is the taxonomy. So let me know once you have done that, just so that I know I'm not losing anyone, because you will actually use this table to understand what's in there. Okay, how many of you have gotten to this place? Okay, once again, so I open this in Excel. Okay, you might have to go to File, Import, and here you do a text file, import, and then you take this table, get data, and it will ask you, how do you want to open it? So how do you want to separate it? And so you can do delimited. And here, you actually want to do tab, and then finish, and okay. So this is how I got the table in. So did everybody get this? Okay. All right, great. Now, okay, maybe in Windows it's a little bit different, but it should be pretty much the same. Okay, so Prerna, so Prerna, you are not able to open it. The table is open. Okay, excellent. All right, so there, Kim is, yeah, if you have an explanation, great. So, you know, this is very basic kind of to open this kind of a table in Excel. We can go over it for somebody that's not so familiar with it, but the basic idea is that once we have this in Excel, and I'm just going to delete this because I already have that. Okay, so once you have it here, you want to just separate the entries that you have here for taxonomy, just copy them over, right? This is where we can just simply go to, you have this menu here and it says data, and in the data you can do text to columns, and in the text to columns you can do delimited, again do next, and here just use you don't have to use tab, just do other, and you do underscore, and it will separate, because you see underscores here, you want to just separate them but treat consecutive delimiters as one, next, okay, and finish. Okay, so this is how we're going to separate it. So for those of you that are following along, please let me know that you got to this point and you have your different levels of taxonomy. So these are levels of taxonomy. Okay, how many people are already here? Just so I know. Okay, Shreya, Prerna. Is anyone else here? Is anyone else? Should I wait a little bit more? Yep. Okay. All right. So I think there's at least 4 people, but there's more people here, so I just wanted to make sure. Okay, so once you have this, you can now make a decision. So you can make a decision by which level of taxonomy do you actually want to organize your data. For example, I've added here, so you can see K is kingdom, P is phylum, C is class, O is order, F is family, G is genus, S is species. As you look at your species, you can see that many of them are missing. If they're missing, that's because there is not that level of annotation in the database. But as you move into the higher levels of taxonomy, you can see that there's going to be more and more annotation. But nevertheless, in each one of them, if, for example, we go and we filter here, you can see that some of them, if I see an F here, that means that some of them are missing. And for each level of taxonomy, I will have, the higher it is, the more I will have full. So this is just important to remember. Here, for example, you can see that also you have a G, right? And so based on this, you can see how many, so here you can see G, 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 right? So some of them are missing. So if they're missing, that means that some of them are just not going to be annotated. So now let's create a new table. So the same as this, but since we want to organize this, we are just going to put here, okay, our level of annotation. For example, if I want to use phylum, I'm just going to take the phylum column, copy it, and I am going to put it right here, and I will just name this by phylum. Okay, did everyone get to this point? So now I have the same data, but instead of my annotation by the ID, I have it organized by phylum. And I'm just going to save this. So my file is called Metagenomics 1 annotation. Okay, I'm just going to save it like that. All right, so let me know once you've got it on your end, so I know to proceed. The second column, this one, this is the abundance. So this is the same column as we have right here. Just here, you can see that this is the result of bow tie annotation file, metagenomics lesson 1, etc. So it actually includes the full kind of information. ERR, remember that's the name of the file. That's the name of our sample. Whenever we got that file from NCBI, this is where it is. Okay, so I just cleaned it up and just left ERR. Okay, so did you get the same thing? Okay, so for those of you that don't have this file and are struggling to create this file, since we're going to be using this file, I'm just going to give you this file so that you can have it. Okay, so this is the actual file. Anyone can download it. Okay, so once we have this file, let's see what's inside. Okay, so what I'm going to do now, I hope all of you had a chance to register here already, but this is a site that we call the Code Playground, and it will simplify some of the things for you as you get started in R and Python. So I encourage all of you to take a look at some of the things that are already open, and we'll add a few more as we go through the program. As we go through the program, we'll start learning how to do a little bit of coding. It's not going to be too much, just simple things here and there, but hopefully you'll get to implement some of these methods that we're using and annotation and visualization of our data so that it's easier to do using these things. Now, what you need to do as you register or as you get here is create a profile using the same email address that you use on the EDU site. Okay, so I know that some of you are already registered, but some of you might not be. So who is already inside here and has a login? So you can see if you're logged in by seeing your email address right here. All right, is anyone else in here? So 2 people are in here. For those of you that are already logged in, go to Playground and we'll use this R Playground. Okay, please let me know once you are in here. Okay. Cam, were you able to make it here? All right, excellent. So I think we have quite a few people. Okay, so what we'll do is go to Playground, right? So go to Playground, and we're going to do a little bit. Okay, so we're going to do just a little bit of coding here just to quickly see what's in our table. Okay, so first of all, what we want to do in R, it's not really complicated for those of you that are not familiar. Obviously, for those of you that are familiar, it's going to be even simpler, but I'll show you where to find some additional tutorials to get this started and eventually onto your computer. Okay, so first of all, we need to have a library that is going to be using the Excel sheet that we have. Okay, so that's the first thing that we're going to put here. Then we're going to load our data. So what we're going to do is we're going to have the actual data. So if we go back to our file, where is our numeric data? Where can I, in my table, where's the numeric data? Right, column B, right? So column B, but without this guy right here, right? So just this is my, right. So this is my numeric data. What I want to do with this data is I want to use it as counts for all of these categories. So let's go back. Oh, let's go back here. And so what we'll do here is we'll call this bacteria count, for example. That's going to be the name. And I'm just going to read table. So I'm going to read. Okay. How about this? Is that better? All right. So read. All right. XLSX. And we're going to put in here the link that I gave you. So that's the file. Now, unfortunately, this becomes a little bit long, but we are just going to take the sheet that is number 1, 2, 3, and 4, right? So this is sheet number 4. So let's take sheet number 4. I don't think that you need to have a GitHub account. Let me just make sure that this is public. Does anyone have this problem? Yeah, you can use your own file, but the thing is that you have to have it from a link because you're using it online. If you are using this, okay, one second, I'm just going to, yeah, this is public. So this is public. What to type after pasting the link? I just do sheet equals 4. So, okay, so let's just do. Okay, so the file is there, right? So we're not going to do this because there's too many, but just to check that it's available. So everybody, and I'm just going to paste this just in case you need to see the exact thing, right? Okay, so again, we'll get to R itself. I wanted to do it here so that we don't have to deal with installation. Some of you might have R installed, some of you might not have R installed, and I don't know who does and who doesn't. So I'm just trying to make sure that we do it in a consistent way. So if you could please follow along with me here, it will save us a lot of back and forth. In your personal R Studio, if you have R Studio, I'll just quickly tell you, you have to go to your files. So you have to go, let's say my file is in downloads. And once I can set this as my working directory. And once you do that, you can just put your name, the name of your file in here. Akil, let me know if that answers your question. Okay, for everyone else, let me just make sure that we get to what we want to do here quickly, and then I'll show you some exercises that you can continue working on after we finish in your own time, maybe over the weekend. Okay, so what do we want to do next? Next, we want to put in just the information from that column, right? So we are going to say, let's create a num table. So this is going to be just our numbers. And we're going to put here the bacteria, we're going to do table and bacteria count. And the column is called ERR1250035. Okay, so this is going to create just the numbers. So let's do table. Okay, so these are now the numbers that we have in our num table. Okay, this is just the numbers. All right, so then we want to add labels, right? So labels, the labels are going to be just the unique values, right? So we can put unique as vector, and then we'll do bacteria count and the name of our column, which is file. Okay, how many of you are here? And let's just do print labels. Okay, use method labels. Okay, in reading the file, are you using this link? So can you paste the error here? Success running code. Okay, well, did you do this print at the end? Divya, did you do print? So do you have all of this? So why don't you try copying and pasting this in there and let me know if you are getting any errors. Now, if you do get an error, tell me exactly, just paste here what the error is. Is anyone, all right, Divya got it? Is anyone else, does anyone else have this? Okay, Shreya, Divya. How about Akil? Did you manage to get that working for you? Okay, and Cam, were you able to do this? Kitty, Michael, Barca? Okay. Sorry to hear that. What I will just say briefly is that all of this information is actually in these two first lessons. So if you go to code.omicslogic.com, you can follow this tutorial and this tutorial, and it will tell you exactly what to do. Okay, anyone else? Is Michael, I don't know, are you got the file? Okay, in your own RStudio, perfect. So you got the file. Okay, so what we want to do is we want to just separate it, the labels from the numbers. Anyone else? Mikhail, I'm assuming you're already far ahead. Barca? Okay, great. So it looks like a few of you are here. Dr. Barca, are you with us here? All right, great. Excellent. So let me know if you have any issues. So once we've got these labels, right, we do want to save how many of these labels we have. So the easy way to do this is to save it in, okay? So if you have any challenges, yeah, just let me know. So how do we actually create a variable? Let's just call it call num. So this is the, or yeah, call num, for example. And we will just do the length of our labels. Okay, so let's just see call num. So we don't need this. Just do print call num. Length 14. Okay, so we have 14 names in there. All right, so the next thing that we want to do is we want to just visualize this, right? So we have our data, we've got 14 unique labels, we've got all kinds of numbers there. Let's see, what are we going to do with this, right? So if we have a total number of variables, right? So here, if we get unique ones, we need to add up all of the numbers that we have, right? And we want to see, for example, which one is the richest, right? So which one is the most abundant? How do we do this? So if we go in Excel, right, and we want to do this in Excel, for example, right, the first thing that we want to do is we want to sum it by the different unique um, names uh, of our labels right here, right? So what we need to do is we want to create a pie chart. Okay, let's say if I have my bacteria, proteobacteria, and bacteroidetes, right? And let's say fusobacteria, and I count them up, and I have 5, 2, and 1, then I can create a pie chart, right? So I can create here a pie chart, and it will look like this. And I will know which one is the most abundant. So proteobacteria is the most abundant. So since we have 14 labels, what we want to do is we want to count them up and summarize in such a way. So the way to do this, I am just going to tell you today how to create a pie chart, but I want you to try and complete these codes right here so that you will know the full code. So today we're just going to do pi, okay, and I'm just going to briefly show you how to create it once you have your summary done on table. We need to take colors, and let's say we can take rainbow colors. And that will be by colnum. So that's the number of colors that we will use. We will use, let's give it a name. So we can call this count or richness. Richness by, this was phylum, right? And C is going to come from our labels. Okay, so if I do this, I will get a pie chart right here. For the different, but I'm just going to already tell you that this is not correct exactly. There are some more things that you need to do, but just based on this, right, you can already see where we are going with this code, right? So the one thing that we need to do is we need to make sure that we are actually getting the right numbers, okay? So this will be your assignment for next time. Next time, we will actually pick it up from here, and we're going to go through code, the full code, and try to compare several different samples between themselves to understand how do we actually measure differences between samples. But up until now, what you should have is you should have the ability to load the file. You should have the ability to count what you have, okay? And by the way, for all of you that, did anyone, by the way, get a chart? All right, great. So if you got a chart, that means something is working. What you want to do is you want to save, and now you have your script on your computer. And what we will do for next time is we'll pick this up from here, and we'll do the full code for multiple samples and how do they compare between themselves, okay? So for those of you that got here, please download the file and save it in a folder. For those of you that do not have had some technical issues, please start going through this through these two exercises. They will give you some basics about setting up. If you don't have R set up on your computer, you can also go to About, And here you will find how to install R and some of the packages that we're going to be using every once in a while. We're not going to really use all of them. You will also have some instructions here about Python. Okay, so just wanted to get us started, see what's inside our data set together. So once we do this, right, once we do this analysis, we can start looking at how different areas in the body, for example, the stomach, right, what we see here is actinobacteria, right? So actinobacteria is typically in a healthy gut the predominant one. And you can see that the second one is probably firmicutes. So this composition is called the normal composition, right? Healthy, so to say. So whenever we have some of our samples that we try to compare between them, we now know that we can look at different levels of taxonomy and look at how composition changes between different samples. Okay? Any questions about what we did today? Or in this first part of today? I have a few more things that I wanted to go through, but are there any questions about what we were trying to do? Okay, so the other thing that we did last time is we looked at how to find data sets from NCBI. And we looked at 3 different types of data that you can find. We looked at bioproject data, SRA, which are the raw sequencing files that you can map onto the reference database and then quantify, or you can actually get some processed data like tables, something that you also already get as a normalized output. Okay, great. So we'll look at that, Dr. Barca. I have a few examples, but before I jump to my examples, let's see if anyone was able to find a data set that was already interesting. So if you did find a data set that is interesting to you, just paste in the link to the data set, and we'll take a look at some of these data sets. Okay, for example, let me give you an example how I would do that for my example. Here, I can go to NCBI. Okay, so let's go to NCBI. And I know that some of you are probably interested in COVID-19. So let's put in here COVID and microbiome. So COVID and microbiome. So we know that COVID is a virus. It enters through the nose and into the lungs. And so we might expect that there is some function for the microbiome related to how the disease progresses. So one of the things that I see here right away is that there's only 3 bioprojects, right? So what are the bioprojects? Bioprojects are actually organized samples according to like a collection. And so you can see here alterations of the human oral and gut microbiome and lipid metabolomics in COVID-19. Here you have no description really, and here gut microbiome alterations and longitudinal kinetics in COVID-19 patients. So if I want to look in here, I would click on this. Here you can see the description a little bit better. We aim to provide proof and evidence of fecal microbiome alterations in COVID-19 and investigated microbiome and SARS-CoV-2 kinetics in 15 diagnosed COVID-19 cases. Okay, looks interesting. So if I want to say, okay, I found this data set and it's interesting to me, I would just take this URL and paste it right here. All right, great. So Cam has this bioproject right here. Let's take a look at it together. The role of snow patches on the spatial distribution of soil microbial communities and biochemical cycling in the Antarctic dry valleys. Samples were collected from dry mineral soils associated with snow patches, which are a significant source of water in this desert environment, at 6 sites located in the major basins of the Taylor and Wright valleys. Okay, great. So what kind of a project is it? What kind of sequencing is this? Right, so Dr. Varka, that's what we are going to look at today, is how to think about what type of analysis could be done with this data. Okay, so Cam is saying this is 156 samples, right? So right there we see 156 samples. And they are primers V6 through V9, right? So there's a total of 9 different hypervariable regions of the 16S ribosomal RNA. So let's see, where is this information? All right, so if we go in here, and we can take one of them, we see here that this is V6 to V9 regions of the 16S ribosomal RNA. And this is soil metagenome, and this is 454 sequencing, metagenomic, and the layout, right? What's important is that this is single read. In other words, as we go into other pipelines that seek to address the challenge of chimeric reads and issues with noise, this is not going to be possible to use. The only way you can actually use single end reads for metagenomic sequencing is by mapping them onto the full 16S ribosomal RNA. Okay, so great. So this is Cam's project. How do you actually download these files? We will take a look at that in a second. How do you know the metadata? What groups, right? So the next question, Cam, maybe you can just think about this if you have this. Identify sample metadata and groups, and think about what kind of information can be extracted from the data set. So this is just for Cam. Let's take a look at Akil's data. So Akil here also pasted a link. Okay, so let's take a look really quickly at Akil's dataset. Next generation sequencing in the diagnosis of viral encephalitis, sensitivity, and clinical limitations. Okay, so Akil, what type of sequencing is this? Okay, Akil, so all of the details, right? So what exactly were they sequencing? What kind of layout, library layout, etc. If you could please list that out. And we'll take a look at Prerna's also. So here we have Hot Springs Metagenome by Bical Rift Zone, 16S Metagenomics of Hydrothermal Vent of Bical Rift Zone. Okay, so Akil, how do you know what is the library layout? You go to, again, SRA experiments, right here, and it tells you it's Illumina HiSeq, right? So it's important to kind of look into this to understand what could be done here. Again, it's metagenomic whole genome sequencing. So when you see Illumina, you know this is shotgun sequencing. And this is viral encephalitis, okay? And this is single reads. So you have to also understand what exactly is the organism, right? Or what are they trying to sequence? Sometimes when they use the word metagenomic sequencing, it could relate to sequencing of viral genomes. It could relate to the whole microbiota, so all of the microorganisms present in a sample. So it could really talk about many different things. Okay, Prerna, let's take a look at yours. So this here says specifically 16S. The moment you see 16S, you know that this is microbial. Microbial because only bacteria have a 16S ribosomal RNA gene. So if we look at SRA experiments, let's see, what do we have here? Again, this is Illumina MiSeq. So when you see Illumina MiSeq, immediately you know that this is probably going to be paired end sequencing. And this is what's called traditional amplicon 16S sequencing. And you can actually read about it right here, okay? So this is going to be, okay. So let's take a look at the last one, Shreya. assessment of interlaboratory variation in the characterization and analysis of the mucosal microbiota in Crohn's disease and ulcerative colitis. Please list out what type of sequencing and exactly what layout are we talking about. Shreya? All right, excellent. Now, let's take a look at Cam. Cam actually has here a full description. So she's saying that there are sample metadata and groups, 39 distinct fields, many held data of limited value, sample depth, elevation, latitude, longitude, which means you can actually map them onto a map, material, soil, organism, sample name, feature, high soil, carbon, conductivity, location, average read length, etc. Okay, so let's take a look together at this project, okay, to understand where do we find all of the needed information. So I'm just going to use this as an example. Again, remember that this is gut microbiome alterations and longitudinal kinetics in COVID-19 patients. So let's take a look at what we have here. If I go to my SRA experiments, Dr. Barker, yes, if the data is whole genome sequencing, you can take these samples and just do what we are doing, mapping them onto the 16S ribosomal RNA gene. If it is amplicon of a specific hypervariable region and it's paired end, there's a little bit more that you can do on the pre-processing side to make sure that your data is going to be denoised, so to say. We'll talk about that in our next lecture. Okay, so here, as you can see, if I just look on the SRAs, it's a little bit difficult to understand what does this data actually talk about. So the way to find all of the relevant metadata is to go to send results to run selector. Here I will have all of my samples run. So these are the names of the samples. And here I have age, right? So this is interesting. We know that, for example, older age is associated with higher risk for COVID-19. I also have sex, female and male, right here, and some other things. Now, typically, if I click on metadata, I should get a little bit more information. So if I click on that metadata, let's take a look at what am I going to find. I'm just going to open this in Excel. As you can see, this is actually comma-separated values. So I'm just going to change the name to CSV. Okay, so here I can also look at isolation source and organism. So there's human gut. All of these are human gut, right? Here I have stool, male and female. So what kinds of comparisons might I be able to do? Do you think? What might I be able to do? Yeah, the metadata is, if you're in the run selector, just right here is metadata. So when you look at this data set, what would you say is a type of a question that I might ask from this data set? What could I compare here to what? Does anyone have any ideas? What, okay, diversity of microbial population, comparison of microbiome by age and sex in the samples, compare gender or age-based. Okay, so how would I do this? And this is kind of like the main issue that we wanted to get at. I want to take a look at my, for example, older and younger, right? So here I have my age column. So what I need is I need obviously the name of the file, right? I need the age. Where's the age? The age is right here. I need the sex, and probably that's it, right? So now I can group them. So for example, if I want to group them by sex, which should be fairly simple, right? I just organize them, and I can compare these versus these. So I have two groups here, male and female. So if I want to do this, how do I actually load up all of these files into my pipeline? If I do all of these, remember we took a look at the Silva database. Who remembers how many sequences are in the Silva database approximately? If we look at even kind of a lower end of taxonomy or a higher end of taxonomy, how many did we see in the Silva in terms of the number of sequences? Remember, there was something like 500,000 sequences. So it's a lot of mapping, right? If we want to take all of our 73 samples and map onto all of the sequences, it's going to take a long time. So instead, if my question is, how do I compare male to female, how would you actually select? What are some of the considerations that you might have to select from each one of these groups that would minimize variation within group to help us study differences between male and female microbiome in COVID-19 infection? How would you go about it? All right, excellent. Dr. Barca suggests that we take the same age, right? So here we have older. So the oldest is about 70. The youngest is about 20. Probably want to have some average. Looks like maybe 48 might be the average. So let's take these three. So we have female right here, right? So I'm going to select these two and maybe another one, 47. Okay, so these I can use for female, and let's find the same approximately from the male. So we don't have exactly many for 48, but here's one, 48. Here's another one, 48, and there's another few 48. So right there. Okay, so if we want to take these samples, we can take them from here. Okay, so we have 9, 13, 14. So we have 9, 13, 14, and we'll take the first one. So these are our male. Oh, okay, never mind. Somehow 14. Oh, okay, we organized them. So let's organize these two by male and female. Forgot about that one. Male and female. Okay, there, host sex. Okay, so which ones did we have here? We had 9, 13, and 14. 9, 13, and 14. So we're just going to put these here. Okay, let's just check. These are all female, great. And for our male, we had 37, 39, and 64. So 37, 39, and 64. 37, 39, and 64. Okay, once I have this table, I can save it. Okay, so I'm actually going to call this male versus female COVID-19 microbiome, and I'm going to use it as TXT, tab delimited text. And now let's build the pipeline together. Okay, so please go with me to server.t-bio.info. Please let me know once you're here. Okay, one more. We're going to this link for anyone who's not. Okay, so we are going here. And instead of clicking on the drop down, we're just going to click on the main portion right here. Is anyone else here following along? Just want to make sure that we're not leaving anyone behind. All right, great. So here we go. And now we need to make some selections about our data. So what do we put in our reference genome? Does anyone have any idea what we, okay, our genome. So in fact, we don't want to use a reference genome at all. We don't know genome right here. All right, great, Michael, got it. Now, is this single end or paired end? Let's go back and take a look, right? So here we have all of our metadata right here. What does it say right here? It says paired end, right? So we need to go to paired end, change it to PE. What kind of what data is it? What type of data are we using? Is it FASTA files or FASTQ? Okay, great. So FASTQ files. And what do we want to map it on? 16S. Perfect. Okay, do not select all bacteria. This is a huge database. All right. So now, how do we upload the data? Click on File Upload. And here, instead of adding the files, we're just going to take directly from NCBI. So I'm going to add files right here. And I called it male versus female COVID-19 microbiome. How many samples did we choose in total? How many samples? Yeah, 6 samples, great. So group selection. Okay, so here's 1, 2, 3, 4, 5, 6, right? So it found them correctly. We're going to do our downstream analysis later. I could organize them by group, but I don't really have a use for it right now. So I just can do cancel no groups. Okay, and then I can build my pipeline. What do I select next? Who remembers? Okay, bow tie. So bow tie to align right here. Then I will do post count. So I'm just going to count and replace the names. Nothing really to change here, and okay, so again, you can now do the same for your data set. What I am showing you here is an example. Does anyone have any questions about how to analyze data from NCBI? Important to remember that when you save, no, it has to be in .txt. So when you download the file, it's in txt. Somehow they save it in txt, but they actually add commas in there. So I don't know what happened on NCBI's end. So that's why we edit it, just to open it in Excel. But if you want to, do the same, so basically it has to be in txt. But here I'm going to say male versus female COVID-19, save, run pipeline. Okay, do not forget to run this pipeline. Now, this pipeline is going to take some time. It's not a process pipeline already, depending on how many samples you have. In the context of this course, please do not use any more than 10 samples, okay? This is not intended for huge data sets. We want to focus on how to analyze, understanding the methods. This is just one of the first methods that we're exploring. There's going to be a few other things that we will do. But for now, please keep it to a small number of samples. Does anyone have any questions about what we did here? Okay, does anyone have any questions about what to do then with the table once you get that table out? No genome, because we're mapping not on a single genome, we're mapping on a database. So we're mapping, remember we have a database called Silva with all the 16S genes, ribosomal RNA genes, and those are helpful because they can help us understand the taxonomy, how they are organized by different levels of taxonomy. These are actually not full genomes, so we don't have a purpose in separating them or mapping them onto a reference genome. Does that make sense, Doctor? Okay, great. All right, so if there are no more questions, I just briefly wanted to show you a few examples. And what kinds of projects, right? So I'm assuming that only a few of you have an idea. Now, don't get stuck on a particular idea that you don't want to, explore other data sets. Many times, as you have seen, you will look into a data set, check its metadata, and we will look at some other examples of data sets as well. You want to select a data set that has a good data set. That good data set means it's got the metadata that could answer your question. For example, if you want to, Dr. Singhal, right, you were asking, where's the control? Where here we don't have a control, right? So how would you compare it to a control? You can't in this specific example. How could you understand whether that person eventually had a lethal outcome or a non-lethal outcome? Again, you cannot answer that question. So as you look through the data sets, make sure that you find data sets that probably have good data, the kind of data you want to use, that have good annotation for that data, and it's in the right format, so you can actually know what type of analysis to use for that data set. So let's take a look at some of these examples, okay? So I saw some of you are looking at environmental samples. Here's an example for healthy gut, healthy adults. The gut microbiome, the important function of the microbiome in the gut is to actually help maintain a certain diversity, so not a single one group dominates the rest. And the reason for that is because they create an optimal environment that both helps metabolize food, extract the nutrients, and maintain a certain level of acidity, which if it changes, a lot of these biochemical processes are also going to be impacted. Now, another function is becoming a firewall, so to say, a level of protection. So they form a biofilm, and this is mucus biofilm that represents the primary barrier that limits contact between the microbiota and host tissue, preventing microbial translocation. So in fact, there's a number of dangerous pathogens that can exist within the gut without causing infection at certain proportions, right? If those proportions increase, all of a sudden they could penetrate this mucous membrane. Now, it's important to understand that a major way that these microbes exist is also contributing to developing certain protection because they really interact with the immune system. So they teach the immune system how to recognize foreign or its own. So really it's a training ground as we grow, as we develop. And so they have an important microbial kind of an immune system contribution as well. And especially in the infant gut bacterial microbiome, there's a process of diversification in the first year of life. Those that typically develop allergies and all kinds of autoimmune diseases like asthma or malnourished, right? So again, this is nutrients coming in. The diversity of those nutrients is very important to create and stabilize a certain environment within the gut. So a lot of these end up in risks for childhood disease. For example, formula feeding, using antibiotics, exposure to all kinds of environmental toxins can have specific impacts on the way this microbiome forms in the first year of childhood. So as a result, this could have a long-term impact. We also have impact on treatment. So for example, anti-cancer therapy a lot of times could be regulated, associated. For example, diarrhea is actually a result of dysbiosis. So we can really see how specific types of probiotics and different supplements can stabilize the way that the microbiome works as a single organ, helping in a lot of cases also absorb the right kind of treatment and eliminate side effects. So there's also a response to treatment that is a factor. We're not going to go into this because we're running out of time. Gut and brain axis, right? So an important connection between the gut and the brain. Those microbes have a way to pass information between the gut and the brain. And so this is an important part of the gut microbiome function as well. And what's important to understand when we are trying to study, for example, inflammation. Inflammation is caused by different agents. Now, we do have those agents under control. And typically, when we look at these studies, one of the immediate things that we can spot is the lack of diversity and some kind of dominance by a particular group of microbes. Now, that dominance or dysbiosis in the regular kind of equilibrium that is established over a long period of time and sustained by proper nutrition could lead to exposure. And so, for example, you can see how in flu, in majority of the cases, this is a lung infection. But sometimes it also causes diarrhea and all kinds of issues in the gut. So that shows you how the importance of healthy microbiome is. And when we take antibiotics, it's important to remember that antibiotics, different types of antibiotics, they actually work to interfere with the process of this ribosomal RNA, so with the ribosome, sorry. And so when they interfere with the function of the ribosome, since the ribosome is not exactly the same for all the different types of bacteria in our gut, oftentimes different types of antibiotics will interact in different ways with our gut microbiome. Once the antibiotics cause dysbiosis and reduce the diversity of the microbiome, we can be exposed and susceptible to different infections that typically would not be affected. Now, a good example, again, I just wanted to kind of highlight that we have this microbiome and COVID-19. I thought that since COVID-19 is an issue now almost everywhere in the world, this could be an interesting topic for everyone. But here we already took a look at some of these data sets, how to find the data set, how to load. There's a good review paper on COVID-19 and the gut microbiome that also touches on a lot of elements of how this works with infectious diseases, with viral pathogens. So you can see here a good paper. I actually posted this paper on the forum already for those of you that might be interested in reading about this. And we went through how to download this data and how to load it up to create a pipeline, okay? Now, I think that we are now ready with kind of getting set up with some of the basics to talk about some of the more advanced analysis that we can do when we want to compare groups of samples. So as you think about the data set that you have selected and you know what to look for, try to find not just any data set. For our next session, we want to only focus on 16S amplicon data. It has to have groups of at least 3 samples each. At least 3 samples in each group, because you need to have some representation for your power of analysis. And it has to be paired end data. Okay, preferably it should be with MySeq Illumina sequencer. Okay, so as you started exploring different data sets, I know you might be interested in all kinds of variable topics, but please try to find specifically a data set that follows these criteria, because we will look at in our next session how to compare diversity between different groups, and we will rely on denoising of that data first. So we will rely on the paired end property of those reads and try to see how to quantify diversity, how to compare diversity between samples. Now, a lot of that information is going to be found in Metagenomics 2. So if you do have time, please try to go through Metagenomics 2. We will explore there the data2 pipeline. It's a full pipeline that includes the phyloseq R package. So we will actually do some more coding in R to know how to visualize, and we'll finish also the first exercise. So we'll look at how to visualize and perform comparison of such data sets. So for next time when we meet, which is going to be September 7th, please try to go through Metagenomics 1 and 2, register, and do the first 2 practices on R in OmicsLogic, that encode OmicsLogic.com. Okay, any questions? Why in R? Well, so data2 is in R. Many other types of analyses rely on some functionality, but of course, yes, you can do, so in Python, you can actually do wrappers of R to run all the same packages if you want. I believe that you have, you know, KIME is a comparable type of pipeline. But we just, for simplicity of everybody being on the same page, we will do it in R together. If we have time at the end, we can also look at how to do this in Python. From bioproject, how much samples can we select for analysis? So please do it under 10. I suggest to keep it simple in the beginning. So let's have 2 groups at least 3, but not more than 5 per group. Okay. So in terms of Python, obviously we will have a tutorial in R and Python next week after everybody has a chance to prepare a data set, right? That's the objective. So Mikhail, I'm actually going to put a lot of effort into making sure that we've got that ready, and we can look at the results and think of what to do in Python as well. Any other questions? No, ion does not. Sorry. Anything else? How is the abundance of read mapping the measure of reliability? Well, the reliability, we'll talk about reliability tomorrow when we look at reliability of our source data. It is actually not so much the abundance of reads, but it is the quality of the read mapping to the reference. So you do have that estimate for each one of your reads. You have a quality measurement for each one of the characters in the read, but you also have the quality of mapping. So there's a number of different types of quality checks that we will talk about next time in the context of the data to pipeline. And we'll look at filtering and removing some of the additional adapters and making sure that our reads are high quality. We will do all of that in R. And we'll also do it on the platform, but we'll also do it in R. Okay, any other questions? All right, excellent. Thank you very much for joining today, and I will see you again on September 7th.